So great. Thanks for inviting me, Chris, and I'm glad to sort of give a testimonial to LabSite. They've been really critical to us being able to get the company off the ground from day one and keep us moving. So I'm going to tell you about what we do at Perlara. We're a patient portal and drug discovery platform for families affected by rare disease. And if you add up all the families affected by rare disease, it's 1 in 10 families on Earth. So let me explain a bit what I mean by a patient portal and a drug discovery platform. Before I do, I'll just note that we're located at QB38953, which I guess is that way, I think. And we're about to raise a Series A and move to our own independent space. We completed several partnerships that have enabled us to move beyond the seed stage. So I'll tell you about the science that has been enabled by the Echo and other automation. So you hear very common themes from the Notable folks. In fact, you can think of us in some ways as kind of what Notable is doing, except instead of focusing on cancer, we're focusing on rare genetic diseases. And instead of looking at directly at patient cells, we're creating patient avatars, in other words, using simple animal models that we can program with CRISPR to carry particular patient genotypes and study them as the model of disease. So we're fundamentally about democratizing drug discovery for rare disease, and the reason I talk about that way is that it's really, everyone knows it's really expensive and really it takes a long time to make a drug if you can even get a candidate all the way through. And so for families that are affected by rare diseases that are as rare as maybe 100 patients in the U.S. or maybe 100 patients globally, known today, the costs and the time is just too daunting. So we're really designed to allow families to go on what we're calling a PerlQuest, which is this early stage drug discovery effort that begins when they complete the so-called diagnostic odyssey, which is the long, arduous process of figuring out what's wrong, which maybe takes 5 to 7 years, 2 to 3 specialists before you figure out that, aha, my child has this particular disease. You end that grueling odyssey, and you now have to begin a new odyssey, which is finding a drug. And we know what the stats are, so we want to make that not so scary, but also make it work, both in terms of science and both in terms of the unit costs. And I'll get into that a little later. But essentially, the patient portal that we've built is really just an MVP. We're inspired, obviously, by our surroundings in tech. We've really just built a simple Google form that says, captures very simple information. Who is a parent or patient advocate that's interested in launching a PerlQuest? What's the disease and what's the gene? And oftentimes, people will just simply share a clinical exome sequencing report with their child's mutations without even us asking. They're so eager to get to jumpstart this process. So starting with this very simple portal, we hope to build a larger community, but essentially this is where we now transition to our automated drug discovery platform. So I'll get back to the PerlQuest in a bit and get back to why this is illustrated and kind of more of the culture and, of the company and the business model. So the drug discovery platform is really at its heart, as I said, about creating patient avatars. So let's take your typical rare genetic disease. There's 4,000 monogenic or single gene diseases. You can imagine a case where you can take a particular mutation and program it into the orthologous position. In other words, this disease gene is oftentimes ancient when you look at these rare diseases. They're not uniquely human genes. They're not uniquely mammalian genes. They're oftentimes ancient genes. And so fish and fruit flies and C. elegans and yeast have orthologs or versions of those disease genes. And oftentimes the amino acid position that's affected in a human being is the same throughout evolution in those simple animals. So when you change that position or break the gene, you're likely to find essentially the same pathophysiology emerging. You can convince yourself that this is the case through doing a natural history study of the disease in these simple animals. And then, of course, once you've got these validated models, you can do drug discovery with them. You could also do gene modifier discovery or biomarker discovery, all manner of things. But we're focusing initially on doing drug discovery with these patient avatars. And the concept here is really to go from zero, essentially creating disease models, to identifying novel lead compounds. Our initial focus is on small molecule drug candidates, and getting to there essentially in a 12 to 15 month cycle time, which is much faster than the typical sort of phase of drug discovery. Usually could be a 3-4 year process, costs just as much money. We want to turn that into less than a year $250,000 proposition, which we think is possible because of the lowered unit economics of using these simple animals. And of course, the lowered unit economics of building a viable drug company. I come from Stuart Schreiber's lab. I did my PhD there, sort of the mecca of chemical biology. And in the early 2000s, if you wanted to build a drug screening facility, that would cost several $1 million, require a well-trained staff, to, essentially 24/7, to maintain the instruments. And it would be impossible for a young company to raise enough money in a seed round or through enough SBIRs to build even the minimum viable drug screening platform. That all changed in the last few years. And really, with 4 instruments, we could build, and essentially $750,000 in CapEx, we could build a minimum viable platform for drug discovery. Very little automation. So not the beautiful automation that was over at Noble we hope to emulate. We have a more complex system where it's not just all a human cell. You're talking about different species, not just 384 plates, 96-fold plates, different kinds of media, fly food, worm food. So it's a little bit harder to create that automation, but we are just in the beginning stages of turning this manual process into one that is modular and scalable. But again, starting with just 4 out-of-the-box instruments, you could build a simple drug discovery platform. So we've partnered our first program in a lysosomal storage disease called Niemann-Pick type C with a big pharma, with Novartis. That deal was announced several months ago. This was really the so-called validating deal. So this was really proving to the world, but also hopefully to families with rare disease, that our platform is legit. And so the PerlQuest model, which I'll get back to, really couldn't be launched without first showing that a big pharma would accept that this model organism-driven platform is a sensible one for rare diseases. So our lead program is focusing on this particular childhood rare disease called Niemann-Pick type C, or NPC disease. To simplify it, it's too much cholesterol in the wrong place. It obviously, even though these are single gene diseases, the NPC1 gene is the affected gene. Single gene disease, but multi-system pathophysiology. Again, it's very glib for me to say too much cholesterol in the wrong place, but that's one way to sort of think about this disease. You can also think about it from the point of view of unmet need. There is no FDA-approved drug, although there are several in trials, and probably cyclodextrin, which is the active ingredient in Febreze, will be successfully repurposed to be used as a disease potential stabilizer for NPC, but not a disease reverser. And a couple of liabilities of cyclodextrin are that it's a very large molecule, so it's not orally bioavailable, and also does not get into the brain. And this is a disease where you need to target not only the liver and spleen, but also the brain. So there's an opportunity for us to find a small molecule that is orally bioavailable, and that does get into the brain, and that operates by a new mechanism, a novel mechanism. And so that's what we set out to do with this program. And it all really started with doing a worm model of Niemann-Pick C disease. So in the simple C. elegans, the simple nematode, there are particular defects associated with Niemann-Pick C disease, quote unquote, in the worm. Obviously, you can't do a complete one-to-one correspondence between how a disease manifests in a simple organism, which doesn't have the organs that go wrong in a human being. But at the molecular level, at the cellular level, you could build a model showing that actually the fundamental events at that level are the same, even though they play out differently at the macroscopic level of organs and tissues. But in the case of the worm, essentially, what is a childhood disease in people is also a childhood disease in a worm, in that you don't actually get developmental timing at the right. You don't have normal developmental timings. In other words, you have stunted development. And even when you get to adulthood, as these worms, they produce fewer progeny that are sicker. And all of these could be phenotypes that work at a macroscopic scale of a plate that could be miniaturized into 3D4 well and done at high throughput. And that's what we did. And so what we set out to do was find a small molecule from a drug-like commercially available library. We set out to find a novel small molecule that would rescue this whole organism phenotype. Most drug discovery is based either on cells or purified targets. No one is doing simple organism drug discovery. Exelixis did this once upon a time, but for a lot of reasons I can say why that didn't work and why it will work now, why it is working now. But effectively, what the screen was designed to do was to say, we know that the worms are developmentally stunted. Could we find a small molecule that could rescue that? And so we have 2 control conditions, the positive control and the negative control. And essentially, you want to make the sick worms in the well on the left look more like the worms on the right. And we found a compound in replicate here that we ended up calling PERL101 that achieved this rescue. We then took this molecule and on the thinking of, well, we want to have a portfolio of disease models. The cell models are usually covered in these cases. The mouse model is usually covered. The problem is in between. And so that's what we've offered to fill in with the flies and the worms. But immediately you need to take a molecule that's active in one system, especially the animal, and move it to a human cell as quickly as possible to understand that the biology you're affecting is conserved. Because you could be off on some worm-specific or invertebrate-specific tangent, and you want to eliminate that as quickly as possible. So the first thing you do is you take PERL 101 and put it on to patient fibroblasts, Niemann-Pick patient fibroblasts that carry the most common patient mutation, which in this case is just denoted as 1061 because it's position amino acid isoleucine change to threonine at that position. And so when we treat these fibroblasts with PERL 101 at 10 micromolar, which is a pretty reasonable screening concentration to look for activity for a primary screening hit, we saw what was actually the counterintuitive phenotype. So normally you would want to see, so let me explain, what you're seeing here is a stain of cholesterol. I said before that Niemann-Pick in a simplification is too much cholesterol in the wrong place. And you can visualize that by looking at the panels on the left, wild-type cells versus the disease cells. You can see the intracellular cholesterol, which is stained by this dye Philippin, shown up here in this blue pattern. In the disease state, you get these accumulations. And the far right is those disease cells treated with SAHA, which is a broad-spectrum HDAC inhibitor, which is an experimental drug that's also being repurposed for Niemann-Pick. And the mechanism here is obviously one of clearance. You're taking this disease state, which is the accumulation of cholesterol, and this compound is causing a clearance state. So that's what we were primed to look for. But this molecule that rescued the worms, when we put them on human fibroblasts with the disease, it caused what looked like the opposite phenotype that we wanted, which is there's more cholesterol, but its distribution was different. And when you looked a little closer with your cell biologist glasses on, you realize there's something interesting going on here. One thing we started to do was understand, well, what is the actual mechanism? We know we couldn't figure out the target just by interrogating these broad phenotypes, but at least we could build a case for what is this molecule doing. And one of the striking features of the molecule is that within minutes of treatment, you would see these tiny uniform vesicles forming, called them bypass vesicles. And we were intrigued to understand what are these molecules, because they were specific to PERL 101. PERL 102 was a compound that also emerged from the worm screen that had the same phenotype in human cells. And so we were intrigued, what were these molecules, what are these vesicles? And we started to do a lot of work over the course of several months, and we narrowed down on a hypothesis that maybe these are actually some kind of lipid droplet or storage droplet for cholesterol and other lipids. And we did a set of experiments looking here at dose response of PERL 101, and we treat with a fluorescent analog of cholesterol in the media. And we ask, what does its uptake look like? And right away, within an hour, this is pretty not illumination compatible for this, but over the course of a few hours, you can start to see these puncta forming. And they are strikingly resembling the puncta that you see just under bright field light. And by 6 hours, you can see this very pronounced effect. And you don't see it in the control condition, which is chloroquine, which is a molecule that we think is actually doing the opposite of what PERL 101 is doing. There's some other cell biology that we get into. We think that this process that PERL 101 is affecting is called lipophagy, which is a kind of autophagy, which is the cells, which was the autophagy, if you don't know, won the Nobel Prize this past year, or the discovery of autophagy in yeast, I might add. And this is a process of self-cannibalization, where cells turn over their components. Lipophagy is a specific form of this, where they turn over lipids. And we can see in this case, in this series here, that cells treated with PERL 101 and stained with this fluorescent cholesterol have this overlap with LC3, which is a marker of autophagy, a classic marker of autophagy. And this overlap is near complete in all cells, which is why you see them orange or yellow. Versus in the chloroquine condition, the control, you don't see any of the fluorescent accumulation of cholesterol. You do see some LC3 accumulation, but there's no overlap. It's all red and green. So this to us suggests that we're able to find compounds with novel mechanism of action using these simple animals, and in a relatively short time identify compounds that we could then put on a trajectory to test in mice. Because the whole goal here is to get to mouse efficacy. And that was the thesis, and was proven because that was what got us basically to get a Pharma deal, was to advance a program to mouse efficacy. That's kind of the point everyone wants to see. But to get to mouse efficacy, it's great if you might have a compound with new efficacy or that has efficacy with a new mechanism. But what if the compound just doesn't have the chops pharmaceutically because it's not orally bioavailable, doesn't get into the brain, the liabilities of something like a cyclodextrin? So what we did is we took PERL 101 and we said, let's figure out if we could take this primary screening hit, do no chemistry, do no lead optimization, and just drive this molecule toward a mouse. And that would prove that if we could essentially obviate the need for hit to lead and even early lead optimization, could this really prove that we've built a platform that, in a faster way, generates higher quality lead compounds? So we put PERL-101 through a barrage of in vitro drug metabolism and stability assays, starting with liver microsomes from mice. And you can see that just asking how much PERL-101 survives over time, we compared several PERLs. So these are the compounds that came from the worm screen and also had this phenotype of this increase in cholesterol and redistribution of cholesterol in human cells. We named those series of compounds PERL. PERL-101 was just the first. And this tells you that not every compound that comes from a worm screen is going to be ideally suited to go to mouse or ideally just suited anywhere. They could be dead ends for the same reasons that a lot of molecules that come from target-based or cell-based screens also fail. But what we think this says is that when you do a screen in whole animals, like a worm, you just select for the possibility of finding compounds with these properties. It doesn't guarantee it. And then when we compared PERL-101 with a bunch of close analogs, it was one of the most stable. So we moved forward with this compound, got it resynthesized, got 20, 30 grams of it, and set on doing a set of mouse experiments. First was, is this molecule better than cyclodextrin from the point of view of, does it get into the brain and can it be given orally? And the answer to both is yes. And I can give you explanations as to why I think what is it about the worm physiology or the conditions of the screen that would select for these PK properties that you would think, how could you select for these in an invertebrate? PERL-101 is well tolerated. We think that's actually another feature of the way we build our screens, because we're taking sick animals and looking for compounds that rescue that. So if you had a compound that was just toxic, you would never see it come up as a hit anyway. But when we took PERL-101 and did a maximum tolerated dose study across several doses, these were just treating wild-type mice over up to 90 days. And we already saw, and these are not Niemann-Pick mutant mice, these are just the wild-type control mice. And we saw a number of interesting things that suggested, well, A, this molecule is clearly not inert. There's some interesting potential effects on body weight gain that we don't really have a good explanation for, but this told us that, hey, this compound really is doing something. And even at an obnoxiously high dose of 120 milligrams per kg per day, these animals survived treatment for over a month at this extremely high exposure. They literally turned orange. The compound is orange, and they literally turned orange from the exposure, but they didn't drop dead. They just started to lose weight and had to be put down. But presumably they would have lost weight on that trajectory. But that weight loss doesn't occur at 80. Even at 60, you see what looks like to be a decelerated weight gain. So something's going on with this molecule, again, testifying to the fact that when you start with whole animals, you get better hits out of the gate. And then finally, we did a set of mouse efficacy studies, and initially focusing on markers of the liver. And we believe we underdosed with PERL-101 because one feature that is maybe a drawback of screening hits that come from whole animals is that they may not be that potent. They may have a lot of other great properties, but for reasons I can get into, they may not be sub-micromolar potency, which is an issue. So we think we underdosed, but even so, we can see that there are effects on liver markers. They approach significance around 0.05. I didn't bother marking that because we think these are simply trending towards significant. We wouldn't stand by these numbers in a court of law, but we believe that the trends are in the right direction, especially when you look only at males. For some reason, the males and the females have a different disease course, and these are Niemann-Pick mutant knockout mice. We don't understand what that sex difference is, but we do know that when you look at the males, the signal gets tighter. And here you see ALT, which is a marker of liver damage. That marker should be elevated in the knockouts. It should go down as you dose higher, and we dose two groups, 20 and 40 mg per kg. And you can see the trend is in the right direction, especially when you look only at males. HDL cholesterol, you can see normalization of those levels. So here's a marker going up. And then with bile acid, this is a log scale, so actually the drop-off in the males is quite impressive. So again, this is not a slam dunk in efficacy because we think we underdose, but the objective here was to take a primary screening hit from a worm and take it to mouse efficacy studies in less than a year to prove that you didn't have to do all that intervening work to get to a compound that's sort of ready to be partnered, because that's essentially, this is the program that we partnered out with Novartis. Now I'll just end by going back to the whole idea of this PerlQuest. So essentially, when we completed this study, we realized, wait a minute, this is essentially a discovery service we could now provide to families, because we have the proof now that a pharma would partner if we were to take an asset, at least to mouse efficacy. So we can now turn around to all the patient groups who had been talking to us over the last few years, wondering if we could work with them and we weren't ready. We can now turn around and say to them, show them this sort of data package and say, this is what we can do. And this became essentially the first PerlQuest. The second PerlQuest, which is really the business model that we're looking at, is about a joint partnership with a patient family. this case, it's with a 501c3, a charity that was formed by a family that's affected by this ultra-rare disease called NGLY1 deficiency, which is right now only has 58 or 60 known patients globally. That number will go up, but right now, less than 100 known kids on planet Earth with this particular disease. And we partnered with this foundation such that they provide the early capital to get this program started. And then in return, they get co-ownership rights of the IP. And we are both incentivized to take an asset as far as we can go. We know if we take it to the mouse stage, we can sell it. We won't get as much as if we took it to phase 1, but we have to spend a lot more to get there. But the idea is that at least if we take it to mouse efficacy, we know, based on the Novartis deal, that we can now go to pharma, not just that pharma, but any pharma, and say, we've got a mouse active compound. This is a program that you guys would buy. And so we essentially started this NGLY1 PerlQuest about a year ago, and it started with, as I referred to in the beginning, a natural history study. So what you do in rare diseases, if you see kids, and it doesn't matter the age, but let's think about kids, if you start basically as a clinician documenting the course of disease as early as you can, and you do that over the course of their life, that's a natural history study when you do this over many, many children. So you could do the same exact study, but with the animal avatar, right? Recreate the disease with CRISPR. You can program in a particular mutation for a particular child if you want, and then you could do a natural history study in the animal. And the animal is obviously going to cooperate because its lifespan is a lot faster and a lot quicker than a human being. You can capture everything about this animal in a way that you can't capture all that data in a person. So that could build the foundation for not only proving that this model is therapeutically relevant to the human case, but once you build essentially this asset, this is co-owned by us, by Perlara, and the patient group. That patient group can now distribute this compound to all its network of academics, and they can go ahead and pursue basic research in tandem to our drug discovery. But what I'll point out here is that when we did this natural history study of NGLY1 deficiency in the fruit fly, we saw that the adults, so essentially a mutant and a sort of a heterozygous control next to it, the mutant animals are smaller as adults. There's other features, if you're a Drosophilist, you might be able to discern, but one striking one was that the animals are smaller. Guess what? The NGLY1 mutant mice are also smaller, and so are the kids, actually. One of the phenotypes is that they're smaller, and they think that's related to some kind of defect in energy expenditure and utilization. So that was nice, and so we wanted to then, of course, understand, is this something that this difference, is this something we could leverage in a high-throughput screen to try to reverse disease in the animal, and then go on from there to mouse efficacy. And so what we ended up showing was that at the larval stage of development, the NGLY1 mutants are also smaller. And so essentially we could build a screen to ask, could we suppress that difference and make the smaller larvae more like their heterozygous siblings in size? And this is just showing you an image, which I think is quite beautiful, of the kind of data that we can generate. And this is with a piece of custom automation we did also with modular science, essentially a custom imager for flies, where we can measure any stage of fly development. This happens to be a particular protocol optimized for larval resolution, but we could also see whole adult animals in here. We could do any stage of development and read it out in a very quantitative way, where we can now go in with software, calculate the size of each animal, and do these very rigorous developmental time courses and milestones in fly, which is exactly the kind of assay we did a year ago or a year and a half ago with worms for NPC. So with that, I'll take questions, and thank you for your attention.